Hi, this is Miss Litton, and this is a review of evolution with my honors biologist. And just a reminder of those of you looking back on this chapter, we have already talked about the steps to life in an earlier chapter, so we're not going to discuss that part again right now. But we were talking about what kinds of things cause evolution, and I asked them just a minute ago before I retaped this, what is something that can cause variations to emerge? DNA mutations. And we'll go through the 5 fingers of evolution. But let's start off with knowing what evolution is, and that's what? A change in, okay, give me another word. Allele frequencies over time. Very good. And the second part we look at is that descent of organisms from a common ancestor. So one of the first things we started talking about in this chapter was evidence for evolution. So those you want to have in your wheelhouse, that would be something on a test where I would say, name and describe 3 different pieces of evidence of evolution. I would expect you to be able to tell me that. So you should be able to know those just from these pictures right here, right? What is this picture about right here? Fossils. So let's do just a little review about fossil. If you wanted to be a fossil, how old do you need to be? At least 10,000 years old. What kind of patterns do we see in the fossil record? When we look back at the geologic time scale and we look at the fossil record, what patterns do we see in fossils? They're going from more simple to more complex. Do you remember that? And we can also see, what do you call it in between one from another? We can see, starts with a T, transitional fossils. We can see transitional forms. So we talked a little bit about fossils. We talked about the way you could be a fossil. What are some different ways you could be a fossil? What are the different types of fossils? You could be petrified. I like it. What else?
Molds and casts.
Molds and casts. What else? Good. Sedimentary rock.
Oh,
Amber.
And ice as well.
And ice. Good job. All right. So we talked about those different kinds of fossils. Then we talked about the geologic time scale. Let's see how well we can remember that. You ready? What era are we in right now? Cenozoic. And this is now, this moment, back to how long ago? Drive your car. 65 million years ago, right? Because we remember driving our car. Now back 65 million years ago is the Cenozoic era. Okay, the era before the Cenozoic era was the Mesozoic in the middle, right? Mesozoic era in the middle. So it ended 65 million years ago because that's when the Cenozoic started, right? 65. And then it goes back in millions of years to what? 248. Good. And the way we remember that is that's about the time that what is out? School. Now, when you think about Mesozoic era, I want you to imagine this picture. You ready? What am I being right now? Dinosaur, and I'm running around a Christmas tree. What are those called? Christmas trees and Hanukkah bushes. What are they called? Gymnosperms. So right in the middle, the Mesozoic, I have dinosaurs running around a Christmas tree, and that'll help you to remember that's when dinosaurs gained ascendancy and also gymnosperms. In the Cenozoic era, we transition from gymnosperms to angiosperms. So hand Angie some flowers. See Angie? She really likes flowers. Here, Angie. Angiosperms, got it? And herbs, right? And humans. Humans came after the dinosaurs. You can remember that, right?
Herbs.
Mammals to humans. Oh, it went from angiosperms to herbs, and it went from early mammals to humans. Just imagine somebody cooking in their kitchen, a human, and adding rosemary to their pasta. That's what I want you to imagine, nothing else. Okay? Stop it. I can see your brain right now. I can still see it. Stop it. I can still see it. Okay? Mesozoic era, what are we thinking about? Dinosaurs running around a Christmas tree, Sam. Okay? Or a Hanukkah bush. Okay? And then before Mesozoic was Old and Paleozoic. So if we have dinosaurs running around a Christmas tree, that's where it ends, right? The beginnings of our dinosaur and the beginnings of our Christmas trees, the beginnings of gymnosperms. So the Paleozoic started back where you had invertebrates. And you work your way from invertebrates to what? What comes after invertebrates? Vertebrates. And then fish, and then amphibians, and then reptiles, the beginnings of reptiles, right? Because they're running around in the Mesozoic era, so it's just the beginnings of them. And then plant-wise, what's the slimy stuff on top of the water, maybe somewhere starts with an A? Algae, and we work our way all the way up to the beginnings of, the beginnings of, what were we running around? Yeah, the gymnosperms. So we go from algae to the beginnings of the Christmas tree. Does that make sense? Okay, then in the middle we have the Christmas tree Hanukkah bush, and to the beginnings of angiosperms, we're handing them to Angie, up to man cooking pasta and putting in rosemary. Got it? Stop it. All right. Before the, when did the Paleozoic era start? 542. What were your 548? 551, 548, and writing that in. And what are we thinking about for that? Dinner. So we remember dinner time, school's out, and driving our car. Now to driving car to school's out, go eat some dinner. Okay? Everything before that is called what? Precambrian. And that's going to start when the Earth started. When did the Earth start? 4.6. When was the first life form? Right. So the first billion years, we were just trying to form a cell. Right? Then the next 3 billion years, life has been evolving. For the next the first billion years was just getting a cell. The next 3 billion was all about life evolving, biological evolution, and together that makes about 4.5 billion years, right? Okay, perfect. All right, so that was the geologic time. That's evidence. We covered those two. What's this one about right here? Biogeographical evidence. And that's saying that as long as the continents are together, this is just one piece of evidence. As long as the continents are together, amphibians, okay, that were in the process of evolving, I'm being a amphibian, they could move around on all the continents, but once the continents were separating from each other, then whoever evolved after the amphibians, right, who was starting to evolve about 65 million years ago?
Humans.
Mammals. So mammals were starting to evolve 65 million years ago, but the continents were separating then. That's why we have unique mammals on all these different continents, but we could have similar amphibians and similar reptiles. But our mammals are very unique to our continents because they evolved and exploited in different environments, and so they have different adaptations. Okay? So that's your biogeographical evidence. And then this one right here is what? Anatomical. Now from anatomical, we had 3 pieces of evidence from anatomical. Do you remember what the 3 pieces were? Homologous structures. What else? Vestigial structures and embryological. Okay? So homologous structures are, hey, you have a humerus, radius, ulna, carpals, metacarpals, phalanges. So since we have this, and you have that, you're a whale, and you have that, you're a fish, and you have that, you're a sloth, and you have that, you're a horse. So since we have similar anatomy, we probably have a common what? Ancestor, common DNA. Those are homologous structures. Now, butterflies and bats both have wings, right? Butterflies and bats both have wings. And so they're flying. That's how they're dealing with their environment. But are they closely related? No, that's called convergent evolution. They just ended up in the same place because they deal with the environment in the same way. Dolphins and whales and tuna and fish, other fish, they all have streamlined bodies. But that's because they're all swimming through water. It doesn't mean that they're related. They're just dealing with their environment in the same way. Does that make sense to you? Okay, those are just called analogous structures. Yes?
Can you list the 3 types of...
Yes. Homologous structures, vestigial structures, and embryological. If you put anatomy right in the middle of it, yeah. Have. But the A is anatomical, right? Have. I have anatomical evidence. Homologous structure, Vestigial, E, embryological. I have that for you. Some anatomical evidence. All right. You're like, that's not helpful. Vestigial structures. What are vestigial structures? I'm an ostrich. What's vestigial on me?
The wing.
Wings. Okay, clearly they can't help me fly now, but it points to my ancestral past. I probably evolved from a bird. Okay, we have several possible vestigial structures. What's this one? Tailbone, right? What were you gonna say?
I was gonna say gluteus maximus.
Yeah, no, but my gluteus maximus is still useful to me now, right?
Yeah.
Yeah, it's still useful to me. So it would be if it wasn't useful to me, right? Okay, and then E, embryological. The more similar embryos are, probably the more closely related, yeah. The last one is the one that is probably most commonly used, especially for closely related species or species, not as long as they're alive now. And the reason is biochemical is you have to have access to their chemicals. You have to have access to DNA, you have to have access to proteins. If you don't have access to those things, then you can't compare DNA sequences or protein sequences or amino acids. I got that? All right. So we did our evidence for evolution, so I'm going. Oh, here's a good one. Okay, so on this one, this is showing you two main types of dating. Relative dating, when you take your cousin to the prom, or when you say the older layers are on the bottom and the newer layers or stratum are on the top. So I'm just assuming age, age in that way. Of course, you'd have to be careful near subduction zones, right? Things like that. Okay. Also, this is an index fossil. It's a trilobite. You only saw it at a certain time, so you can use it. And if there's a fossil layer below that, it's older than 540 million years. If you see a fossil layer above it, it's new, right? Absolute dating is different. Absolute dating would be, let me get a picture. Okay. Stop it. Okay, so in us, as long as I'm alive, I'm going to be assimilating carbon in my body, right? Because I'm going to eat. Am I going to eat carbohydrates? Yes. Okay, so I have a certain ratio of carbon 12 to carbon 14. That's not proportional, okay? That's not accurate. It just makes it easier, right? So I know how much carbon 12 to carbon 14 I would find in something living today. So if I go and find a fossil, and my fossil has proportionally this much carbon 14, which looks like about half as much as what I would have today, then I know this thing, because I know this carbon 12 is stable, Mabel, okay? It's not breaking down, but this carbon 14 right here is unstable, and it wants to break down to a more stable element. So I know how fast it breaks down. So if I know how fast it breaks down, I refer to it when half of it breaks down, that would be a what? Half-life. So I know the half-life for carbon 14 is 5,730 years, so that's how old this is. You see what I mean? Because half of it is broken down. If I found another, fossil, okay, and it had about this much carbon 14 based to what I know exists today, this looks like another half-life is, ratio-wise, it's probably been another half-life. So now it's 5,007, 5,007, right? Now I have two half-lives have passed. Right? Does that make sense? Okay? And if I found one that was only this big, I would know another half-life had passed. And if I found one that only had half of that, I know another half-life has passed. So you're just comparing the ratio today of carbon 14 to carbon 12 to what exists right now. And based on the proportion, because this is the thing that's unstable. Now, I can't use carbon 14 for something that's, 50 million years old, because by then all the carbon 14 will have broken down. So I'm going to have to pick a different element that has a longer half-life of time. So you have to choose the right isotope for what you're trying to measure. Okay, and this is referred to Not as relative dating, but what? Absolute dating. Then we look at this, and we already reviewed that, right? Totally reviewed that. No problem. And we reviewed all of that, reviewed all of that, reviewed all of that. Oh, so then we talked about cladograms. Remember cladograms? So we said, okay, this right here, this spot, this red, is a common ancestor to all of these guys right here, right? At this point, this is a common ancestor to all of them. But this right here, this one, is a common ancestor to all of these. Why? Because this ancestor up here had jaws and hagfish doesn't. And if I said this one right, oh, whoops, not that. This one right here, this is a common ancestor to these guys right here. Because they, from here on up, they all had claws. So cladograms, you say, what's something that has all of the traits we're looking at, right? Then we're going to put that down here towards the bottom. Then you might have some outgroup. You maybe have some invertebrate, something that doesn't have any of those things you're looking at. And then you start going, okay, well, from here on up, they have this. From here on up, they have jaws. From here on up, they have lungs. And then we keep eliminating, and the way you sort it, you don't have something showing up and then disappearing and then re-showing up amongst some other ones. You want to sort it where it makes the most sense logically. So you have you want to try to make it monophyletic, so there's just one single branch working your way up. Yes?
Are we going to need to know like how to like make one of these for the test?
I might have you organize. It'd be very simple.
Okay.
Yeah, but I could have you organize something like that. Yeah. Like in the written part. Might give you a chance to practice with nails. Yeah, because so here, like I have different baggies of nails, and they're all different types. So decide who's the ancestral nail and who evolved from who. It's really fun to do it with candy bars, and then eat them, right? Because you're like, maybe the chocolate kiss is the ancestor. And then you add in, well, this one has some filling in it. So from here on up, they have filling. From here on up, you know, or from, you see what I'm saying? You can do all kinds of things with that. Yes?
If we have time at the end of the review, can you make one like together? Because
Sure.
I'm just practicing. This is one of the things I'm just like.
Well, I have one that's already in here. Do you remember how to do this one?
I kind of got it, but it's
Well,
just confusing.
well, let's do this one because it's right in here. Okay? So I could have arranged these in any order. So let's, if we looked at hair, do fish have hair?
No.
Does salamander have hair? No. Does a lizard have hair? Does a mouse have hair?
Yes.
Does an ape have hair?
Yes.
Does a human have hair? Yes. Okay, let's look at lungs. Do fish have lungs? Salamander have lungs? Yes. Does a lizard have lungs? Yes. These all do, right? So something that's found in more things like this, like lungs, is going to be down closer to the bottom, right? How many up here have thumbs? Does a fish have thumbs? Salamander. Lizard? Mouse? Ape? Human? Yes. So that's something where fewer organisms have it. You'll go, okay, if I was going to do a cladogram just based on this, based on this, maybe my outgroup here is a fish. Okay, here's my fish. He doesn't get to be in any of our club games because he doesn't have all the things we need. Okay, but from here on up, what do they all have in here? They all have what? Lungs, right? So everybody up here has lungs. What's the next one we're going to use? What? Hair. Right, just the three we looked at. I'm just trying to make it like, how would we go about doing it? So they all have what? Hair. Do they all have hair? No, but that's the next most common. We all said lungs, right? So we're going to say from here, you have to have hair. So who doesn't get to be in our group anymore? Who doesn't have hair? Who doesn't have it? The lizard doesn't have it. Okay, so we'll put the lizard. Of the ones we talked about, who doesn't have it? Oh, salamander and lizard. That's fine. We'll just put lizard. Because I didn't do it for all of them. Okay? Then what's the next thing we're going to talk about that they all have? Thumbs. Okay, so I'm going to put thumbs right here. But who do I have to leave out now? Who doesn't have thumbs? The mouse. So I leave the mouse out. Do you see what I'm saying?
Yeah.
That's all you do is you go, okay, so here everybody has this, okay? But then you just start going, well, now I'm going to make the requirements to be in my team be this. Now I'm going to make my requirements in my team to be this. And you want to just keep leaving one off each time.
Like a diagonal, like greatest.
Yes, yes. Most traits, fewer traits. Okay? All right, so then we talked about cladograms, and then we started talking about evolution being a result of 4 factors. So one is, if I'm going to spread out into, if I'm going to change, there has to be room for my change, right? I have to have room to expand. I have to be able to beat out the competition that are currently living there, right? I have to be able to compete and win if my trait's going to get passed on. Okay, so I either have to be better than the current organisms, or somehow the current organisms got wiped out and now there's room for me. Okay, then there have to be variations within our DNA. Those variations have to be able to get passed on. It has to be a heritable variation. So I can't work out really hard in my lifetime and then my offspring are all going to be strong as a result of that, right? Because they're not going to inherit that. It has to be some variation that's in my DNA. Maybe I just have bigger muscles and it's in my DNA. That's something I can pass on to my offspring. Okay? And the best adapted one will win. And we talked about microevolution versus macroevolution. And we looked at the moths and who was best adapted in these different environments here. And we talked about microevolution with this yellow-bellied three-toed skink and how he's keeping his embryos inside. So if we were going to make a cladogram, we might go able to retain embryos. You know, and he gets to, but his relatives cannot, right? And so that's microevolution. If he stops interbreeding with those that can't keep him inside, then he forms a new what? Species. And we're not doing microevolution, we're doing macroevolution. Good job. Okay, then we talked about the 5 fingers of evolution. That's a good video to go back and watch. Okay, and then we brought it into Hardy-Weinberg, these 2 equations.
This is what I also done.
Okay, that's what I'm going to go over right now. Okay, so in Hardy-Weinberg, you have 2 things here. Okay, so let me go back. I have, here we go. Okay, so here is my gene pool. Whoa, I want to be a pin. Here is my gene pool right here. Okay, what are my 2 options in my gene pool? Big B and little b. So I can be big B plus little b is going to equal 100% of my gene pool. Do you agree with that? Mm-hmm. Where did this gene pool come from? Well, this guy threw in a big B and he threw in a what? Little b. So those 2 came from him. What would he be? He'd be a PQ, right? Because he has a big B and a little b, right? This one threw in 2 big B's. Do you see that? So there are his 2 B. What would he have been? A P squared, right? This one threw in 2 little b's into our gene pool. What would he be?
Q squared.
Q squared. So in our general population, this is what you can be. You can be a P squared, or you can be a PQ, or you can be a PQ, or you can be a QP, right? Either way, it's the same thing. So that's why you just say 2PQ. Because you can be a PQ or a QP, but either way, you're a different one, right? So I can be a P squared, a 2PQ, or I can be a Q squared. And that's going to be 100% of all these animals out here. So this is referring to these guys out here. I'm either 2 big B's, I'm big B, little b, or little b, big B. That's why there's a 2 here, right? Either way, it turns out the same. And that's why we can put 2 and not write PQ plus QP. And then this one, I'm little b, little b. And this equals 100% of all these animals here. If each of these animals threw their 2 alleles, whatever they are, if they threw their alleles in here, that's the gene pool, and that's the big B plus little b equals 100%, or another way to write that is P plus Q equals 1. So if I told you, let's say with those wild boars, let's say I told you P equals 0.7. could you tell me how many Boers were big B, big B, little b, and little b, little b, if I told you there were 200 Boers? I want to know how many are big B, little b, and little b. Remember, this is going to be Q squared. This is going to be your 2PQs, because I can be big B, little b, or little b, big B, same thing. And this is going to be what? P squared. So if I told you P equals 0.7, what does Q have to be?
0.3.
Yeah, Q has to be 0.3, because my P's and Q's all have to add up to 100% of my gene pool, and that would all add up to 1, right? So if I knew that, I could predict how many of the Boers would be P squared. What do I have to do? 0.7 times what? 0.7 times how many Boers do I have? 200. So that would be 18, carry the 1, 98? Does that sound about right? Do the math.
98, yeah.
98, okay. Then I want to know how many of them are 2PQs. Well, that would be big B, little b, or little b, big B. That would be the two ways to be it. So that would be 2 times 0.7 times 0.3 times 200 Boers. 84, right. Okay, and if I want to know how many, I could just take these and add them together and subtract them from 200, or I could actually do the math for it. So that would be what? 0.3 times 0.3 times 200. 18. Okay. So do you see how we worked it from here over? Or I could have just given you this number right here. I could have just given you, hey, there's 18 little b's. Oh, you would have gone backwards like this. If I had given you that, you would have said, oh, then 18 divided by 200, right? You would say that's what percent? 18 divided by 200. Find out.
That would equal
0.09. Okay? And that equals Q squared, right? Little b. So that means if I take the square root of it, then Q equals what? 0.3. You see, I can work it forward or I can work it backwards. I can't do it via the dominant traits because when I look at the dominant traits, I've got two variables. I've got P's and I've got Q's. You can't solve when you have two variables, right, and get a final number, right?
Just say hi.
Hi, Fran.
I haven't seen you, so I just came to say hi today.
Hi.
Hi.
You look super cute.
Well, thanks. I was going for that.
I like it. You got the little
Yeah.
skier.
I like your sweater.
I know.
Good
Super
night.
cute.
I hope everything's like settled down.
I hope so. We have a lot of fires here in Cali.
Ugh. All right. Have a good night.
All right. Question.
Can we do another one?
Sure. Let's do one I did with 0 period, and I'm going to do it with you tomorrow in class. Let's do it in this room right now. There's 1, 2, 3, 4, 5, 6, 7. We're going to count her. 8, 9. 9 of us. 9 of us in this room. Okay? I want to know how many of you can roll your tongues like this. Is there anybody that can't roll their tongue? Okay. She is recessive. That's a recessive trait. So who is she? She's Q squared. So 1 of our 9 is Q squared. What is 1 divided by 9? See, you already understand this from next year.
0.1.
What is it? 0.111? Okay, how are we going to solve for Q?
Square root.
Okay, do it. Don't square it, square root it. What's the square root of 0.11?
It's 0.3316624.
0.3 what?
It's just 0.3, I guess. 0.33.
Okay, 0.33. Well, if Q is 0.33, right, what do we know P is if Q is 0.33? Is that right?
Yep.
Do you agree with that? So if I wanted to know how many of you are heterozygous for this, okay, what would I do? How would I find out the heterozygotes?
0.6
No. Heterozygous, PQs.
Okay.
Yeah, so it's P squareds plus 2PQs, right? Because it could be PQ or QP, so we just say 2PQ. These can roll, okay? Plus Q squared equals 100%, right, or 1. So these are our non-rollers, okay? So I have solved now for Q, which means I can solve for P, because P plus Q equals 1. So my heterozygotes, the ones that are carriers for the non-rolling trait who could have children that are non-rolling, that would be 2 times what? 0.33 times 9, because there's 9 of us in this room.
That would actually be 4.0 because it's 3.9.
So what is it? 4?
Yeah, 4.
Okay, so 4 of us in this room are, statistically speaking, we're heterozygotes for it, and we could have non-rolling children. Okay, so 4 of us are here, 1 of us is here. What does that mean P squared has to be? 4. Let's see if that's true. Let's see if that's true. P squared would be what? 0.67 times 0.67 times 9.
Yeah, 4.
Okay, there we did it. How do you feel about that? Okay, so evolution is if this changed. Right now, our P is 0.67. What if our P changes? It'll be interesting in class tomorrow to see what it is. We need to remember what this is. Let's say P became 0.8. What would that mean Q has to be? 0.2. If our allele frequencies change, what is occurring? Evolution. What are the 5 ways that could happen? If our populations are small, if we have non-random mating, if we have mutations, if we have gene flow, and if we have adaptations. Since we can't maintain those things can happen, we are more likely to evolve. Oh, look, now she's focused on it. Let me ask you a question. This small population, okay, the reason why small populations can cause changes in allele frequencies, small populations, is because they are more populations. They are more impacted, small populations, by what? What? Chance. Chance impacts small populations more than large populations. We said there were 2 types, and we refer to when chance does that to small populations, that's called genetic drift. There were 2 types of genetic drift. Do you remember what they were?
Gene.
2 types of genetic drift.
Is it
Founder's effect. Yeah.
What's the difference between the...
Probably I'm going to explain that. Okay. So bottleneck effect, okay, that's when there's some disaster that wipes out part of your small population, so only a small bit start. And is this, our S and M, representative of all the letters here? No. Okay, so that is a bottleneck when a crisis wipes some out. Okay, example of founder's effect would be if these 2 letters decide to go to an island and start their own population over here. They didn't represent the whole team here. They just represented N and S. Does that make sense? Okay, so that is not a good representative because we have P's and L's. Look, we have a lot of L's. Like we have 3 L's. So they didn't even represent the L's. Okay, so that would be, those are the 2 forms of genetic drift. That help? Mm-hmm. Okay, and then was there any, let me just glance. Oh, 2 forms of speciation, best sum, well, 3 actually, right? Slate, oh, sorry. This is a river runs through it and they evolve, go down their own pathways. What kind of speciation is that called? A barrier, it starts with a barrier. What's that called? Allopatric. See the barrier? Oh. Allopatric. Okay? And then this, when it happens right beside each other, what is that? Sympatric. We're right beside each other. We're very similar. We were right by each other, and we just changed. You changed on me. That's sympatric speciation. And then the last one, if it goes into a whole bunch of different species of trees out from a single hub, what's that one called? Adaptive radiation. If this happens really slowly, it's called phyletic gradualism. If it happens really quickly, it's called punctuated. Yep, you did it. Chapter 27. Good job. I'm super proud of you. You're welcome. Have a piece of toast. Yeah, read this last slide. This is what can happen as a result. Okay, that's a good